Okay, good afternoon, everyone again. For those who just joined us, welcome to our faculty panel on promoting student inquiry in science and engineering lab courses. I'm John Fu, Assistant Director for Science and Engineering on our Faculty Programs and Services team here at the Center for Teaching and Learning. And joining me today is my colleague Saika Asuda, our Programs Coordinator, and she'll be providing support during our panel. And so before I introduce our panelists and begin our panel, I'd first like to ask you to sign in. Saika has shared the link in the chat. We'll use this list to send our follow-up email with additional resources. And you also have the opportunity to sign up for our weekly faculty newsletter where we share upcoming programs and new resources. Just a brief overview of our session today. This will consist of two parts. For the first part, each of our panelists will give a short presentation on their lab course and the opportunities they've incorporated for their students to ask their own questions, make their own decisions, generate their own interpretations, pursue their interests, or demonstrate autonomy in some other way. And during the presentations, everyone will be muted because we're recording this. And after the presentations, we will have a Q&A session with our panel. You're welcome to begin chatting your questions during the presentations as well as they arise. And we can definitely circle back to them during the Q&A session. But during the Q&A session itself, we will allow you to unmute your mic so that you can also ask your questions directly if you'd like. And now to introduce our panelists. Joining us today are Dr. Sarah Hansen, Senior Lecturer in the discipline of Chemistry, Dr. Aaron Kyle, Senior Lecturer in the discipline of Engineering Design in the Department of Biomedical Engineering, and Dr. Terryanne Maenza-Gmelch, Senior Lecturer in Environmental Science at Barnard. As you can see, our panelists today are from different disciplines. They have different courses with different learning objectives for their students. So we'll get a sense of a whole variety of ways that we might be able to gain inspiration in how we can incorporate opportunities for students to have more inquiry in our lab courses. And so without further delay, I'm going to invite Dr. Sarah Hansen to present first. Sarah?
Thank you very much. So I'm just going to go ahead and share my screen. Welcome. I'm so glad to see turnout here for this topic that's really, it's dear to my heart and really important for our students on campus. Whatever they're studying, welcoming them into our labs is really a key experience during their studies at Columbia and Barnard. So I teach the general chemistry lab. This class it's kind of a classic in a lot of different programs and series, and possibly the beginning of the lab series that many students will experience on their ways to other disciplines and other labs. And so this class is really focused around teaching how to work in a chemistry lab, how to design and execute experiments, and how to collaborate and problem solve. So we focus a lot on techniques. We focus a lot on safety, good cultures of safety, waste disposal, how to properly use and when you need different types of PPE, how to move about a lab, how to collaborate in a team, what makes a good bench space or an organization, a lot of skills that really transfer to other research and actually other learning environments. But we focus them around chemistry investigations. This course, not only do we do some experimental design and analysis, but we're also asking students to evaluate the reliability of their results and make claims, not that they successfully carried out the experiment, but they were able to actually evaluate what aspects of the experiment, procedural, technical, the equipment they use, or the methodology they choose, especially for the analysis, that's impacting that reliability of their results. And so in the fall, we have about 40 students, 40 to 60 students. In the spring, we get a lot larger, about 120 students. And then in the summer, we have about 40 students. And those are just the sections that I teach. And so we do have multiple instructors, and you can double all of those numbers if you want to look at our total impact of this course on campus. Here are just two images from different experiments. On the left, these test tubes, this image was taken in spring of 2020, and this experiment is still part of our course. But one thing that we've done with the new design since the pandemic hit is to replace a lot of those test tubes that were just being used and disposed of with well plates, and keeping the same concepts, but with new techniques, how to clean and reuse a well plate, but also less impact on our resources. On the right is one of our new experiments developed with the Provost grant, and we just did it last week. It's using oxidation reduction potentiometers in order to do a redox titration and introduce just the beginning of titration and reading and basically pulling data off of a graph. And so we start out with that, and then we build into acid-base titrations in the next experiment where things get a little bit more complicated. Well, what inspired the changes that we currently see in the course? And it's a constantly evolving course. I started teaching this course in 2004 at Columbia, and I am never tired of it. There's always something new and a new way to adapt the course. And as we all know, adapting lab courses for the pandemic and teaching online was an experience. So these two images are two Zoom shots used with permission from students who took the Zoom course. On the left, you can see a student setup for their practical evaluation. Students had autonomy. They had to adapt. And the reason they had to do that is because not everyone had the same supplies. Some students took the lab during lockdown and they were not able to go out and get different supplies. So we ended up with a really rich experience of students bringing what they had available and being able to talk, have a really substantive conversation about what mattered, then connected all of our supplies that we brought so that we could do filtration. So talking about the pore size of the filter paper and what they used for a filter paper and how we evaluate if this technique is working. And all of that had to be done without an instructor physically present in the room for a student. And so on the left, you can see the student has set up their camera so that we can supervise them, so they can work, so they feel supported and they get feedback, but they also develop autonomy on their own. And students told us that they really gained confidence by literally having that space to work. On the right is a kind of a combination of red cabbage juice using, looking at different, using it as an indicator for Alka-Seltzer experiments. And then there's also a casein isolation going on the cloth up at the top. And so this is an example of our final projects where students actually designed an investigation within parameters that were given to keep them safe and to make sure that it fit with what we were doing in the class. And the feedback on that was just incredible. They really enjoyed the opportunity to take charge, to make decisions, even with constraints that were given to them. They still really enjoyed the freedom to have a voice in authoring their investigation and determining what would be an appropriate data analysis and safety measures. They had to come up with a safety plan and actually carry it out. So this fall in the lab, here's an example of a new experiment. Instead of giving students a cookbook procedure, what we do now is we actually, we give them some hints, we give them some tools. We have a whole lecture, about an hour-long lecture every Monday. And then parts of the experiment are authored by students. So here's an investigation where they're trying to find the volume of aluminum in a drink can without opening the can. They had to write a procedure for how they would do that. They had samples of different sodas in beakers, so they could sample a soda and find the density. And then they had to go pick out which can was theirs. And I didn't tell them that it would be floating in a container. But when they came up, they were trying to decide, well, I have a darker colored solution or I have a more clear solution. So I can kind of determine if it's going to be the Sprite or the Pepsi. But then when they came up, they actually saw that the diet or those with less sugar in them were floating. And I will say that now that we're in person, I was really thankful to have a mask on that day because I was smiling so big, seeing students delighted to make the decision and being able to talk to each other. Instead of every experiment kind of happening separately, but in parallel, students were turning to each other and asking what their density was. Students were asking, well, how did you decide to solve this problem and approach it? And so students are individually creating a draft of their procedure, collaboratively deciding on it with their group, and then checking with each other as well as the TA before proceeding. So it's really exciting as an instructor to see the shift that's occurred when we give students that space to design their own experiments. And you can see this one is just experiment 2. We have aspects of this in all of our experiments 1 through 9 leading up to 10, which is just a blank scaffold, and they're going to fill in the entire thing. The other aspect of this course that I do think is really critical is developing technical skills. And so students during the pandemic would watch the TA demonstrate a technique, and they would collaboratively create a Google Doc. On that Google Doc, they wrote the key tips for doing that technique. So here's an example of students' tips that they wrote out for pipetting. The TA would then edit that or add any comments if anything was missing. Students would record a technique video and critique themselves. As long as their critique is complete, they get full credit. And this we have kept from the pandemic into our in-person teaching. And it's going to culminate in a practical evaluation where students will actually be using MediaThread, thanks to the support from CTL, to annotate their own practical and essentially grade themselves with this, me and the TAs actually checking that and providing feedback. So the goal here is to give students the opportunities to develop their self-critique of their technical skills, of their course design, of their experimental design, and how they work in a group. We even asked them to evaluate, how was your bench space set up? What could have been done better? What would you like to think about for next time? And then in the next experiment, after we do that one, we ask them to evaluate how they collaborated in a group, what aspects of group work were key to successful interaction, and what they'd like to do differently or ask of their partners. So aspects of self-critique I found really, really useful and honestly really exciting, hearing how students jump right in to taking charge of the learning process. And so the last aspect I just wanted to mention is not just self-critique, but they actually have opportunities to critique how they take exams. When they take the exam, they know right up front that they're going to have an opportunity to go back and earn some partial credit by evaluating how they wrote out their answers and creating new questions. And so we've added this aspect of the course into not only the experimental design, how they engage with the course and the course space and each other, their techniques, but then even how they take exams and asking them to reflect and get points back on exams. So thank you for that. I'll pass it on to the next speaker. And if there's any questions, I look forward to them.
Thank you so much, Sarah. It was really great to hear that some of the creativity that occurred during the pandemic with having to move things online, that you're able to keep them in the course moving forward. And so up next, we have Dr. Erin Kyle. Erin.
Thank you, John, but also not thank you so much. You put me in a bad spot because that was awesome. I don't know if I can live up to that, but I'm going to try. That was so cool. All right, let me share my screen here. Okay. All right, so once again, thank you, CTL, for inviting me to chat with everyone today. I'm going to talk to you a bit about a course that I developed here at Columbia that focuses on bioinstrumentation. And it's going to be more weighted towards kind of what is the overarching structure of the course, with a little bit of discussion about what we did in response to the pandemic. But I guess I'm becoming increasingly optimistic that we can take some of the things we learned and maybe not have to go back to that so much, but just be kind of incorporating, like Sarah so eloquently pointed out. So I teach lab courses in the Department of Biomedical Engineering, and I've been teaching some permutation of instrumentation since I was a graduate student. And one of the common issues we bump up against is kind of where or what sort of things are our students learning. And the easiest way to look at this is with respect to Bloom's taxonomy, which I'm sure we're all very well familiar with. And the thing that I've commonly bumped up against in the teaching of laboratory courses is that we do pretty well at the basal level. Students are able to remember the concepts and remember some of the steps we're taking. But as engineers, it really is important for us to get to the apex of what we're doing here, to be able to design things, to really be able to evaluate solutions, to be able to iteratively redefine and redesign what we're doing in terms of prior findings. So with that in mind, in 2013, I started working with a group of graduate students in the Department of Biomedical Engineering at Columbia, and we all had seen kind of some of the shortcomings that our students were bumping up against with regards to instrumentation and electrical engineering skills, and we thought we'd develop a new course. And initially, it did look like it was just going to be kind of this structured lab lecture course and kind of a conventional pathway to help students kind of bolster the experiences they were getting in other classes. But as we really start to examine what was going wrong here and why students weren't reaching kind of that pinnacle of learning with respect to the taxonomy, we recognized that there was an opportunity to do something different. So we kind of came up with a different model for our instrumentation course. And we were fortunate back in 2013 to get some funding from MathWorks to help us bring this class to fruition. So what we created was this class called Biomedical Engineering 4740, which is cleverly titled Bioinstrumentation. And the objectives that you see for the course are listed on this slide here. So this really is a design-centric course. That is, we want to make sure that our students not only have a fundamental understanding of circuits, signal processing, signal acquisition, microcontroller programming that go into creating a biomedical device, but also have the capacity to design, test, and redesign based on the outcomes of their system performance. And in addition to hitting all of these objectives that are very design focused, we thought one of the more interesting ways that we could go about imparting these concepts to students was through the development of a medical device. Now, obviously, the things that we do in the classroom cannot meet the level of rigor that might come into an FDA-approved device, but also a lot of things that we work with that are diagnostics or therapeutics or even preventative measures can be decomposed into a few fairly simple blocks that allow for exploration and development within the classroom environment. So we chose to focus on what is my favorite biomedical device, and that's truly a testament to my science and engineering nerddom that I have a favorite device, the cardiac pacemaker. And for those of you that are not familiar with pacemakers, it's actually pretty simple in terms of what they do. A pacemaker is essentially a device that senses cardiac electrical activity and responds in a stimulatory fashion to any sort of abnormal event. The simplest abnormal event we can bump into with a pacemaker is bradycardia, an abnormally slow heart rate. So what we decided to do was decompose the pacemaker into its fundamental blocks, that is, being able to sense, being able to acquire and process, and finally be able to stimulate tissues in response to an arrhythmic event. So over the course of our class, we charge our students with learning about these concepts from the standpoints of anatomy and electrophysiology, and then applying them to the creation of our benchtop pacemaker. Within our first module, we focus heavily on the creation of analog circuitry for the purpose of being able to acquire a good quality version of the cardiac electrical signal that will inform our pacemaker's performance. In our second module, students have to develop the A to D conversion system that will allow for faithful reproduction of the signals in the digital domain. Our third module focuses on kind of algorithmic thinking and computational thinking for the purposes of developing algorithms that will be responsive to arrhythmic events. Now, during our fourth module is when we start to tie this back into a living system. That is, we didn't want to just develop a pacemaker that would work theoretically. We wanted to come up with something that could work with cardiac tissues. So we use a frog as our model system. And the great thing about frogs, besides them being extremely cute and fairly pleasant animals, is that you can knock out the central nervous system of a frog and the cardiac system will function for quite a bit of time. I've had frog hearts remain beating for up to 7 or 8 hours after destroying their brains and their spinal columns. So we do this double pithing procedure. We do a thoracic dissection, and on the still beating heart, we are able to test out the quality of our electrical signal measurement apparatus that we have developed throughout the course. And we characterize the frog myocardium to determine what are the optimal parameters in terms of intensity and duration for stimulating these cardiac tissues. Again, mimicking a pacemaker. A pacemaker doesn't provide this huge jolt of energy in response to an arrhythmic event. It's a fairly small stimulus that propagates through the normal processes of cardiac tissues for the purposes of eliciting a contraction. So we give the students a chance to do some hands-on kind of wet work with regards to working with frogs. Our fifth module focuses on creating the requisite stimulation circuitry for administering electrical energy to the tissue. So we don't use commercial stimulators, we use BJTs for the purposes of applying this electrical energy to our subjects. Throughout the course of the semester, our students are building up these different blocks of the system, but we don't simply build these things in isolation and discard them. Instead, in order to embrace that spirit of iterative redesign and being able to evaluate the whole system, each of the blocks are meant to come together in the last few weeks of the semester to show that they work in terms of the entire sequence. We revisit our frogs to show that we can both sense, process, and stimulate tissues in response to arrhythmic events. And finally, we charge our students with identifying some additional functionality beyond the basal operation of a pacemaker, so they can add something that would be clinically relevant and of their own design to these systems. This has proven to be a fairly successful course, not to be self-aggrandizing, but our students oftentimes emphasize the amount of learning and understanding that comes about from this class, especially those that have prior experience with circuits. This really does allow them to solidify their understanding of concepts, which oftentimes are somewhat evanescent with our biomedical engineering students. In terms of indirect assessment, course quality and amount learned routinely ranks greater than 4.5 in terms of student reporting since its implementation in 2014. We've learned a good deal about this course. In fact, we were published in the IEEE transactions on education for the development and implementation of this course. I can send anyone the citation if they're interested. But we have developed what, to our mind, is a novel paradigm for teaching instrumentation in this kind of design framework. We're pretty pleased with the diversity that this encourages. And what I mean by diversity is the students' backgrounds. That is, we have a lot of students who that aren't even engineering majors, that have never taken an instrumentation course. We have a good kind of breakdown of students in terms of gender balance within our classroom, giving people the opportunity to be more hands-on in doing these sorts of things. And again, this has facilitated some pretty interesting educational research. With regards to the pandemic, like everyone else, we had to shut down in March of 2020. We were somewhat fortunate in that it fell at a point in class where much of our hands-on work had already been done, but we did have to pivot to more of an online model. So our final two modules, which at that time were focused on our Arduino programming, as well as creating our stimulation circuitry, lent themselves pretty well to online simulations of the circuit. So we worked primarily with Tinkercad as our mechanisms for emulating Arduino performance, as well as the implementation of our stimulation circuitry. I had always been a little wary of video recording lectures because if for no other reason, it's kind of embarrassing when you make a mistake in front of people and it's recorded for posterity's sake. But I have come to embrace the video lecture. Students have told me repeatedly that this helps me out a lot. It's good for me to be able to go back and look at things at my own pace, to be able to review things. It still kind of creeps me out when I look at the coursework stats on the videos and I see someone who's binge watched me like I'm Bridgerton, but it's okay if it works out for the students in terms of learning. But we've taken some of those things and incorporated them into being back in person. So we actually were back in person in the spring, albeit in kind of a reduced capacity. I would have one student from each group coming in and performing the labs each week. We progressively increased to 2 as vaccination rates increased. All of our lectures were online via Zoom, again, making sure that we video recorded everything. But it lends itself well to, again, kind of the sort of inquiry, the question and answer, and still having the design activities. I did distill a lot of the content down to what was most important, since I would have one person instead of 4 working on each one of the laboratory procedures. But in terms of the students self-reporting on their experiences, it did not deviate all that much from what we had seen previously. Students were still quite pleased with the quality as well as the amount learned during the spring 2020 offering of the virtual course. So in terms of what's next, this course is offered annually. In fact, I'm teaching it again this fall. We normally only teach it in the spring, but I taught it spring, fall this year. We continue to be interested in dissemination. I've shared this content with other like-minded instructors throughout the country. And one of the beautiful things about this model is that it can probably be translated to other biomedical devices, particularly sensing stimulatory devices. I've got this vision with enough time that I'll be able to transition to another device that I think is really cool, the cochlear implant, which is, again, fundamentally something that senses, in this case, a pressure signal, converts into electrical signal that stimulates neuronal tissues for the purposes of processing sound. So I think a lot of the things that we are doing with the pacemaker can be readily translated to a new project theme. So with that, I conclude. I thank you again for your time and attention, and I welcome any questions or comments about what we're doing in bioinstrumentation.
Thank you so much for that presentation, Aaron. I love seeing how throughout the course, they're basically building up to that pacemaker. So they're designing it every step of the way. So thank you for sharing that. And finally, we have Dr. Terryanne Maenza-Gmelch. Terryanne.
Okay. Well, thank you. I appreciate being here. And these are very exciting talks and from such different backgrounds, chem and biomedicine. And now I'm going to take you into the field. I'm doing Intro to Environmental Science lecture and some labs at the Environmental Science Department at Barnard College. And this is an undergraduate course. The lab course I'm going to talk about is connected to this big lecture course, which has 120 undergraduates. The weekly labs, they have about 16 to 18 students each, and there are 7 lab sections. So they're broken into these smaller groups, which allows us to do some pretty cool things, I think. We generally examine the basic principles of environmental science using current local and global environmental news as case studies. So we're constantly reading current periodicals to see what's hot to talk about, and then we learn the science behind those issues. And so these topics range from things like climate change, biodiversity, ecosystems, water resources, environmental justice, pollution, energy, sustainability. It's quite a collection of topics. So since when we're in person, we're right near the Hudson River, we focus on the Hudson River as our study site, and of course its forested shorelines. And since we go out in most labs, we have a lot of hands-on inquiry-based field experiences where students collect data and then analyze and present on those data. And within the lab, we then start looking at statistics. We look at water chemistry, microbiology of the Hudson River, the microscopic and macroscopic life in the river. And then we move onto land and look at birds and plants in Riverside Park, biodiversity on rooftops, local geology, carbon sequestration in campus trees. And we learn how to use maps and museum resources to help, bolster our work. Basically, what we do is I supply a bunch of readings with focus questions to get students deep into the topics. And then I ask them to kind of formulate predictions or hypotheses about the topics. And then we, as groups, all of the labs go out over the course of the week and collect data. And then we pull the data together. And then the students can then apply their predictions and hypotheses to this pool of data. So they analyze it and then they write it up in the format of a scientific paper. So here is an example of students at the Harlem Pierce Park collecting water samples. And so even though they're broken into the 7 labs, all 120 students will collect data, and we'll have a big data set on Hudson River salinity, dissolved oxygen, and bacteria. And that becomes available for everyone to, ask questions about. We also go out into Riverside Park and do bird surveys in relation to different environmental variables. We'll measure a bunch of different things, and students can then compare whatever they like. So for example, some students might look at bird species richness in relation to canopy density of the trees, or maybe they might look at bird species richness in relation to distance from the busy street, from the street to deeper parts of the park. It's basically up to them. Here's an example of students guided, by the lab instructors, which would be me, and then Professor Sedalia Rodriguez also teaches some of the labs. They get familiar with tree identification so that they can actually participate meaningfully in the data collection. Here are some students actually collecting some of the data. So they're measuring using DBH tapes to get diameters of trees. And then we put that all into a big Excel file. And then students could ask a couple of different questions depending on what their interest is. They could do a size class analysis of the trees in the park, or maybe they might calculate carbon storage in the trees in the park, or even the ones on the campus or on the street trees. There are a lot of options. Here's a student that's surveying plants up on the green roof on top of the Diana Center at Barnard's campus. And they typically can do biodiversity calculations with this information, which then leads to a couple of different other questions. For example, do the invasive species take over? You know, that could be a prediction, and they could actually answer that question by doing this kind of work. So then, of course, you know, pandemic comes, and I can no longer go out with students to our local green spaces like the Hudson River and Riverside Park, Morningside Park, the local geology, the green roofs. I had students attending, just like all of you other professors, attending from all over the place. I particularly noted that I had students from Israel, LA, Brooklyn, New York, New Jersey, Germany, Italy, Cambodia, Singapore. It was quite a group. And I immediately thought, I personally would love to know where everyone's drinking water is coming from, how their sewage is treated, what's their biodiversity like, how's the pollution situation going on there. So immediately it became student use of their local green spaces and experiences for a focus for the remote learning that we had in fall 2020. So this basically equaled an opportunity for students to share their firsthand local observations for a truly authentic global perspective for the entire class to enjoy. And people tuned in. Attendance was very high. It was like almost like a Netflix series, tuning into something interesting twice a week. So here's an example of one of the remote things that we did. I asked the students to find their local shoreline or waterway, whether it be a stream or a river, and then walk along that shoreline for 10 minutes and make observations. And they could make observations about anything they wanted. Turned out that a lot of them made observations about trash that they saw because it was easily quantified. So once I saw that popular input, I decided to have them use a shared Google Sheet and enter all the data. And so we then have a product such as this showing global shoreline trash. And of course, the plastic bottles are number one, right? Another thing was I prompted students to examine the contents of their clothes closet or their dresser to formulate some kind of an analysis. Some of them decided to categorize fabrics and their impact on the environment, or maybe they reflected on where they obtained their clothes. And I also always ask students to include selfies or pictures of their local waterways or biodiversity. So I just put a couple of those into this talk. Some of them chose to actually calculate the carbon footprint of their wardrobe using some math models that are available. And that turned out to be very interesting. And then lastly, well, this is just a picture of what I had told you about before. I asked them to go to their local waterways, and they would report from their local waterway via a 60-second video, and they would post it to the lab site, and we'd all get to watch everyone's 60-second video as a way to be introduced to these local habitats. And so that's it. I'd just like to sum up with that back in person, I would love to continue with the application of the class concepts to personal experiences and their local resources. It's very effective to increase engagement. We have a lot of non-majors in the course. So out of the 120 students, currently I have like 35 first years, 35 sophomores, 35 juniors, and about 15 seniors. Most of them are non-majors, some of them are potential majors. So I think this is an engaging method. And then the last part I'd leave with is that always ask the students to connect the topic to their hometown or state or their own neighborhood, just to increase that engagement. And that's it.
Thank you so much, Terryanne. It's so wonderful to see like there's so much around us that students can work with, and that having students also co-collect data and having this large data set that they could ask their own questions about. I think that's really fantastic idea. And I see that Jamie had mentioned in the chat that the shoreline water observation is really fascinating. What a great way to capitalize on local settings and gather global data. So thank you so much for sharing all of that. And so thank you again to all three panelists for their insightful presentations. Now we'll begin the Q&A portion of the panel. So we are allowing you to unmute yourselves now. So feel free to chime in with any questions that you want, either again by unmuting yourselves, or you can also put it in the chat. But before we go on to those questions, one question I'm just going to ask all three panelists right now is, when you were incorporating these opportunities for students to have more inquiry and more autonomy in your course, did you face any challenges? And if so, how did you address them?
I mean, I have an answer for that. I think you have to scaffold it. If you just give them very broad, like go out and look at something type of a situation, it can be a little bit unwieldy. Oftentimes you would have to have many one-to-one meetings to make sure you could kind of reel them in so that they can make a testable question. So providing a set of readings and a topic, and then asking them to think about it and pose a question, and then collecting data will potentially answer most of these questions that'll come up is a good way to keep it under control.
Yeah, I think I agree that the scaffolding is an excellent point. I also find that that a bit of iteration helps out, or a lot of iteration helps out. Oftentimes I'll have somebody approach with something that maybe is too broad or too much to be accomplished within the project's frameworks, but usually you can kind of decompose it into something that's a bit more manageable or a bit more realistic. Like a lot of times, say like with the pacemaker projects and the students putting on an extra functionality, it might be something that it's like, oh, well, this would be cool. It's like, well, yeah, but what's the relevance of that in terms of an actual pacemaker? And then if we, oftentimes though, that thing that they're interested in, there might be some sort of tie-in to what's really there. So being encouraging of those interests, instead of just saying, oh, that's not right, do something else, but instead saying, let's see if we can find something that is appropriate for the device that we are considering.
Yeah, and I'd add to that, I totally agree, scaffolding and iterative. And then because I'm teaching an intro lab, I found it very helpful to model the first one. So the first lab on the electronic lab manual is the same for every experiment. So it's very iterative, it's scaffolded with parts, but then we give them quite a bit of the first one and even the Google Doc. And then we start taking it away slowly, but it's the same thing. And it's the same, that iterative component and that scaffolded component, I agree, it's absolutely key.
Thank you so much for sharing that. Yeah, I'm so glad to hear all the different ways that you're supporting students throughout their growth in this process. And so I'm going to open it up to our audience for any other questions.
I had a quick question. So thank you all for the wonderful presentations. I love how hands-on these lab courses are and how they're structured to really promote student agency and everything that the students engage in. And I wanted to ask if Dr. Kyle could elaborate a little more on encouraging student diversity, because I think it's really important to try to draw students who are non-engineering majors and other STEM fields, and really encourage them to explore disciplines that may not be within their comfort zone. Because I hear so many, I don't have the brain for engineering or other STEM fields. So how do you approach this kind of perception that some students might have, and how do you approach increasing the student diversity in these courses?
Sure, I greatly appreciate that question, because it is very important, right? It's a way of thinking about diversity beyond the way we traditionally look at diverse issues here, right? There's diversity of background and interest. And what I've learned over the years of teaching, well, I'll giving a bit of background here. I mean, when we developed this course, my motivation was to have a course to augment my undergraduate's instrumentation knowledge. But what it's become, or who are more interested in it, quite frankly, are our master's students. And our master's program is quite diverse in terms of our students' background, because a lot of them do come from basic sciences. A lot of them have never taken engineering or engineering courses. And when we first started that, I thought it was an interesting little kind of quirk in the system that I hadn't anticipated. But it's over the years that I've come to appreciate it, because in our course, it is a lot of what I would consider remediation. It's a lot of, all right, you might have seen this in your introductory physics course or a circuits course that you've taken before, but it's a safe bet you've seen some version of this before. And we try to couple that. We try to couple the reiteration, or in some cases, the introduction of foundational concepts with the application of it, which is kind of my bread and butter as an educator. I'm almost always focusing on, let's get the concept with the application as soon as we can. And by doing that, it's become what I think is a pretty welcoming environment to people of these different backgrounds. I get, every time I offer the course, there's always somebody that says, oh, I've never taken a programming course, or I've never taken a circuits course. Am I going to be okay for this? And the thing I always say is, don't worry about prereqs and things like that. We're going to go through things slowly, explain them in adequate detail. If you need, we'll get together offline. That's one of the kind of few perks of this last year, is how adept we've all become at setting up the Zoom meeting when we might have a couple of extra minutes at home or things like that, and being able to talk through things in sometimes exhaustive detail. But I've become really appreciative of this opportunity to work with people that are not from engineering backgrounds, and to be able to introduce this mindset a bit more, and have them think along these lines. And again, even some of the ones that are from engineering backgrounds, I mean, sometimes they've come from places where there's been a severe gender imbalance in their classrooms, which is common within engineering. And within biomedical engineering at Columbia, it is more of the sort of, assuming a binary, it is more of a closer to a 50-50 split. And this gives a lot of the young ladies in my class an opportunity to do this hands-on work that they might have missed during their undergraduate study or previous courses. And that's the sort of thing I don't so explicitly emphasize, but I'm glad it's an opportunity that presents itself. I hope that sufficiently addresses your question. It's basically kind of a side effect of the course I developed, which has made me quite happy.
Oh yeah, thank you. And I just chatted too. I really appreciate your emphasis on this, your remark about creating this welcoming environment for all students, because I think that does really make a difference in how these courses are presented to students of different backgrounds.
I once tried to get a student in the School of Continuing Studies to join us, because I told her about the stuff we did with frogs and things like that, and she seemed kind of creeped out by it, but I explained to her how interesting it was, and she said she had never done anything. I tried, I didn't quite convince her, but I think she would have had a good experience. I think honestly, almost anybody can take on this stuff to different levels.
Thank you so much, Jamie, for your question, and to Aaron for that wonderful explanation. Any other questions from the audience?
I have a question. I'm Brian May. I'm actually in Terryanne's department. I have a question about this. How much do you let the students fail along the way? Like, when do you finally catch them? And then how do you react to it?
Well, I can speak very quickly about my course, because I took up a lot of oxygen in that last answer. The nice thing about the fact that all of these blocks that we're developing contribute throughout the semester means that we don't necessarily have to get absolutely done within 1 week. Yes, there are certain questions within the laboratory procedure that you would hope they get the data they need to address it. Another kind of positive benefit of the pandemic is that I've got data sets for basically all my lab procedures. So if people need to, I can give them a backup set that's of exemplary performance. But what we emphasize is, you might have done it this way when you first saw it, but based on what you see now, go back and re-examine it, redesign it. Maybe it didn't work. You got another few weeks to get it done. We've got a pretty good chunk of time at the end of the semester too, where we just focus on bringing everything together. So that allows us the opportunity to try different things out and to make mistakes, and to try some other things out. It particularly comes into play when we have our kind of end of semester project, and people might try out 2 or 3 different things that don't work so well.
I appreciate that you mentioned that. In Gen Chem Lab, usually you have to get this experiment done, and then we move on to the next experiment, the next experiment. And when we had a paradigm where everyone did everything and you had to jump through these hoops in order to finish the experiment, and everyone was kind of independently working in their groups, so I guess collaborative, but very small groups. it was really hard to share data. It was more like I can borrow data from another group. But the more that we've shifted to, and I hear this in the other speakers, is kind of group work and collaborative approaches, so that if you understand what's happening and you're able to discuss it, it's not that you're getting the data from another group, but we're actually increasing our data set by looking at all of this collaboratively. And that has become incredibly key this semester. Not a lot, but a couple students have had to Zoom into the lab because they're in quarantine. And as long as they're involved in the entire conversation, we can do the techniques another time, they can still be part of that conversation. That's something I didn't plan for, but has worked out really well with shifting the focus from you must do everything to you must be involved in all of the conversations, but there's no reason why everyone has to collect 10 trials. If one person collects 3 trials but understands the process, we can really focus on doing it together and more collaborative approaches.
And I guess I just have a quick response to Brian, and I'd like to pull in what Sarah said earlier, that in the beginning, you give a lot of scaffolding up front, and then you slowly take away. And sometimes the students that are even at the point when you're giving a lot, some of the students are still not quite able to handle that. So what I do is just on a one-to-one, I continue to give them more scaffolding and take it away at a slower rate, just to kind of work individually with that student. And that's a little bit of a challenge because there are 120 students in the class, so it's not perfect, but that's generally what I try to do.
There's some really neat research in productive struggle that has inspired my comfort level in letting students do the wrong thing. I had students reacting Alka-Seltzer in a graduated cylinder, and it worked eventually, but it takes a long time because it's a tall column versus a larger surface area. And the instructor in me wants to jump in and talk them through how to redesign that. And instead, me and the TAs, we supported them, kept it safe, but let them get to the conclusion that next time I might do it differently, and this is how it might impact my data. But I wouldn't call that a fail, but more a very, very productive struggle. If we had tried to provide all the answers there, they would have lost that opportunity to really reflect. And honestly, their data was fine. It just took a lot longer.
Just to Sarah's point, so everyone's always interested in grades, like what grade am I going to get in this class, right? So if someone has a productive struggle, that could also earn big points as opposed to just getting it right straight off the bat. I mean, I think maybe everyone might agree, right? Or no?
I think that's a great point, because there is a focus on grades. And the more we can remind students that we're creating a safe space for them to take risks, and it won't necessarily impact their grade if they make the wrong decision or they talk to another student or they collaborate. I think it creates the environment that I'm hoping we can foster in our labs and foster in science in general.
Yeah, I also kind of approach it from the standpoint of when we're in the classroom, this is a good place to make mistakes. People are generally friendly with each other, want to help each other out. And I always say, I'd rather you make your mistake here than be out on your first job or be at a conference or something like that and make the mistake in front of everybody else. So hopefully that fosters a sense of comfort within the community that we're building.
Thank you so much for all those responses. I think it's wonderful to hear about how firstly you're creating that safe environment for students to be making those mistakes or having that productive struggle, and building in both opportunities and time for them to do so. And then also possibly rewarding that growth that students exhibit as a result of that productive struggle. So thank you. And thank you to Brian for asking that question. We probably have time for just 1 or 2 more questions. Would someone else like to ask a question?
Well, I actually have a question for Sarah, because I don't know when I might bump into you again. I'm really curious about the stuff you mentioned about the students critiquing each other, as well as the potential for reviewing their process on examinations. I mean, especially if you're talking about awarding points back, what is speaking to that horrible question of grades, what's kind of the basis for saying, oh, this is a good critique, or this is a reasonable critique, or yeah, I agree with you, how you found you had made some mistake in your process, and what would be the basis for giving stuff back? I've always had this pie in the sky idea of having my students like write up the protocols for the labs or review each other's lab reports. And I guess I haven't been brave enough to put it into practice.
It has been very exciting because my biggest worry was students wouldn't want to videotape themselves, and they had no issue with it as long as they knew that only they and the TA would see it, and the TA wasn't grading their technique. So it lowered the bar tremendously for being able to engage with a pipette or engage with a burette and be able to try something. But we grade their techniques in the before the practical. The technique is simply graded on, is the critique complete and is it accurate? And I believe it's like 4 or 5 points out of a 40-point lab report. They get a couple points just for having the video attached, and then a couple points for accurately evaluating. So if they said, with their pipette, if they raised to eye level and they said they didn't, they lose some accuracy points. If they did raise to eye level and they said that they did, then they get those points. And then as long as they've taken all of the bulleted points that the group co-created in the Google Doc, saying these are the things that we need to do to properly pipette, then they get the full credit. So it's honestly, somebody could do a technique where they say, yep, oh, I need to raise to eye level. I didn't raise it to eye level. I need to transfer, but I spilled it. They could take all of the tips and put them in the I need to improve column. They'd get full credit. The key is that they are identifying what they need to focus on for the practical evaluation. And the part that I found really exciting is instead of going to a book from American Chemical Society of how do you properly pipette and giving them what we say is important, the students are telling us what they think is important in their own words. And oftentimes I find some of their descriptions are awesome. They're really descriptive. They're kind of hilarious at times, the way they relate in a somewhat non-scientific vocabulary, but they're complete. They do an excellent job of critiquing the very first time they see a new technique. I've been incredibly impressed. I'm always impressed with our students, but how much they just engage, especially when we take off that, if you don't do it perfectly, I'm going to take off points. They still take it very seriously. And then there's one piece I didn't add to the practical. If you get below a certain point, then we meet one-on-one and make sure you can do it. And I'm a little embarrassed to say that in almost 20 years of teaching, it's only once they were on their own on Zoom that I absolutely had a mechanism to make sure everyone could do every technique. It just wasn't something we assumed that we were implicitly teaching this stuff. And now we are very explicitly teaching it. So yeah, it's been really great. I have a question also. Can I come visit your classes? And actually for anyone who's attending this, one of the things I found most helpful is to just basically swing by a class or listen to how classes go, because the dynamic is so different. I would love to visit these other 2 classes and just hear how it goes.
We're doing frog work on Friday, October 29th. It's always a fun day to come by.
And we're doing birds and plants in Riverside Park this week, 1:10 p.m. every day, if you're interested.
I will send you an email because I would really love to just hear the dynamic of how students engage with these concepts. I've been really impressed by how much our classes have in common at its heart, even if the disciplines manifest them a little bit differently.
And thank you all for being so generous and inviting others to observe your class. Completely agree. Being there in person is a really wonderful way to see that happen and see the wonderful things that students are doing and the conversations that they're having. So thank you so much again for all your generosity. And with that, we've come to the end of our panel. A big thank you again to all our panelists for taking the time to share their experiences and insights with us today. And thank you to the audience for joining us today as well. We hope this panel sparked ideas for you that you can use to promote student inquiry in your own lab courses. Before you leave, I'd like to ask you to take a 1-minute survey to fill our engagement survey that Sayaka shared in the chat. We'd love to hear what takeaways you have from this session, as well as for other teaching and learning scenarios you'd like the CTL to address in future programming and resources. Thank you so much again, and we look forward to seeing you at a future event.
Thank you.
Thank you.